Hi, Mike Gibson coming to you live from AHA 2023, and we are talking about the genetic treatment of hyperlipidemia. Very exciting information. Uh, Sake and Andrew, talk to us a little bit about the unmet need, which I think is pretty clear in this area, and what you did.
Mike, Verve is founded on a simple research insight. If one's blood LDL cholesterol is very low lifelong, it's really hard to get a heart attack. Now, gene editing is a new technology that could be a one-time treatment leading to permanent lowering of LDL cholesterol. And specifically, in vivo base editing, which is what we're using, allows us to potentially make just a single spelling change in the DNA of the liver to turn off a cholesterol raising gene. And that's what we're trying to do.
And specifically, that gene was the PCSK9 gene in this instance.
That's exactly right. What we did, Mike, was develop an investigational medicine called VERVE-101 to turn off a cholesterol raising gene, PCSK9, in the liver. Now, we previously tested this medicine in cells and in non-human primates and shown that it can work and is well tolerated. Now, in the study that's presented on Sunday called Heart-1, we've tested this medicine, Mike, in humans for the first time. We've treated 10 patients with familial hypercholesterolemia, as you know, a genetic disease characterized by very high LDL cholesterol starting at birth and premature heart attacks running in families. Each patient got a one-time intravenous infusion of the gene editing medicine VERVE-101.
Wow, so one and done is the philosophy. So, Andrew, what did you find?
Well, we found that the drug worked the way we thought it would. We were able to see dose-dependent reductions in blood LDL cholesterol after administration. In the 3 patients who got the 2 highest dose levels, we saw LDL-C reductions after just 1 month of 39%, 48%, and 55% when compared to baseline. In the single patient at the highest dose, where we have the longest follow-up, the 55% reduction in LDL has been durable already to 6 months after just 1 IV treatment. This is, in fact, the first evidence that one can successfully change a single DNA base pair letter in the liver in a human being for a clinical effect.
Just amazing. So, what are some of the next steps? This is an intermediate report from a phase 1 study. So, what are you planning on next?
Well, as you say, it's a small study. We've only dosed 10 patients so far. So the next step is to dose more patients. The safety profile we've seen supports the continued development, and we plan to dose more patients at the 2 higher dose levels and build out that evidence base that we just saw preliminary data on. Ultimately, we do plan to test the drug in large randomized controlled trials to fully demonstrate the risk-benefit profile.
So, tell me about the future. I mean, where do you see this ending up? Do you have other drugs into development that would follow the same path?
Well,
We Go
go
ahead.
ahead, Sig.
We do. We actually have 2 additional products. The first one targets PCSK9, of course. The second one targets ANGPTL3, and the third targets the gene Lp(a). These are 3 different axes of risk for heart disease, and we ultimately intend to have a gene editing product against each one of these axes. You know, what we've shown here, Mike, is that gene editing technology can work in humans to turn off a cholesterol raising gene in the liver, and really raising the possibility of a one-time treatment leading to permanent lowering of LDL cholesterol.
Just amazing. I mean, I have to be honest, never thought we would see this in my lifetime. For people like me who have an elevated Lp(a), we're anxiously awaiting treatments like this. Thanks to both of you for this exciting data. I think it's going to be one of the highlights here at AHA. And thanks to the audience for joining us here today.
Thank you, Mike.
Thank you.